Hey guys, it's Taylor West with the Exam 2 video for immunology. So these are some of the things that we're going to be discussing today. Our topics are endogenous versus exogenous, which is we're just going to discuss the pathways for those and what comes along with those things. Then we'll be discussing cross-presentation of exogenous antigens. So this will just show how endogenous and exogenous pathways can cross between each other. Also, we will discuss the presentation of non-peptide antigens. We will compare and contrast MHCs. So first we have endogenous. Endogenous antigens are those that are generated within the cell. So these antigens are already within the cell. So this can be processed in the endogenous pathway. So in this endogenous pathway, the antigen meets the proteasome and moves into the rough excuse me, rough endoplasmic reticulum, and this is where if they bind properly to the MHC, the peptide will move out of the rough endoplasmic reticulum and move to the Golgi body and come out and be presented on the cell. Um, these are presented with class 1 MHCs, so this is very important. The antigens are degraded by the proteasome. So this proteasome, as you can see right here, is linked to ubiquitin. So this breaks down the protein. This converts the proteins into smaller peptides. Next, we have exogenous. The exogenous antigens are taken up from extracellular environment by endocytosis. So these antigens are not inside the cell already. These were taken from the outside. So this is the exogenous pathway. These are presented on class 2 molecules instead of class 1. The difference between these is MHC class 2 can leave the rough endoplasmic reticulum before antigens have been inserted into their groove. So unlike the endogenous pathway, the peptide had to bond and then it moved out. But this one, the MHC class 2 can move out to the Golgi body and come down, and when the exogenous antigen takes up those peptides, they can bind to the MHC and then present itself on the molecule. So extracellular antigens are engulfed into endocytic compartments where they are degraded by acid. So this is very important. So that's the difference between the two. So endogenous is MHC class 1 and exogenous is MHC class 2. So now we have cross presentation of exogenous antigens. Antigen-presenting cells will divert antigens obtained by endocytosis, which we previously stated was exogenous antigens. So endocytosis is exogenous antigens. So these antigens will divert to a pathway that leads to class 1 MHC loading and peptide presentation to cytotoxic T cells. So this will be like the endogenous pathway. So you are crossing over both pathways. What would usually be presented by class 2 MHCs becomes redirected and is now presented by MHC1 peptide. So these two pathways are crossing over, and you're taking this exogenous antigen that's been taken up from extracellular environments and putting it with the endogenous pathway, so with the MHC class 1. Dendritic cells are primary cross-presenting cells. Most potent reside in secondary lymphoid organs where they are believed to receive antigens from extracellular sources by handoff from migrating antigen-presenting cells or from dying infected cells. So what that means is the dendritic cells can play a part in passing these peptides from one pathway to another. Okay, so next we have the presentation of non-peptide antigens. So there are various types of T cells that can react against lipid antigens, such as glycolic acid derived from Mycobacterium. So this is very important for these non-peptide antigens. These antigens are presented by CD1 families. So CD1 family is structural to MHC class 1s, but functional to MHC class 2s. This process, so this presentation of non-peptide antigens, this process relies primarily on hydrophobic interactions that can be expressed on these molecules. So the CD1 family is formed by transmembrane heavy chain composed of 3 extracellular alpha domains, which associates non-covalently with a beta-2 microglobin. So we have these 3 sections, so A, C, F, and T. So glycolipid, which are short chain lipids, antigens, they are this lipid fits into deep pockets within the CD1 binding groove, and the hydrophobic head group remains exposed, allowing recognition by T cells. So this pocket right here, the T pocket, some of these pockets correspond with antigen binding pockets of MHC1, allowing antigens to access a deep groove through a narrow opening, kind of like a foot sliding into a shoe. So the shoe right here is your T binding site. This serves as a binding site on an MHC for a non-peptide. So it's more hydrophobic interactions that can be expressed on these molecules. So the antigen will slide in here like your foot in a shoe, and if it fits, it will still stick up to be recognized by T cells. So like kind of like your ankle sticking up out of your shoe. So this will be able to be recognized by T cells. So your A, C, and F are your deep binding pockets, but your T is your deep binding tunnel. Okay, so now we have MHCs. MHCs are very important. So MHC1, which we have right here, which is mostly white because it's one peptide, react with cytotoxic T cells because the antigen comes from the inside of the cell. So that's important because that's the difference between the two. The MHC1 has the antigen inside the cell. So these have very small antigens that are representing antigens found endogenously, in that cell. So this little red section right here is the short antigen, little chain of antigen that we have inside this MHC. For MHC2, we have a blue section and a white section. This shows that there are 2 different peptides. There is a longer groove for MHC2. That shows that this groove allows for longer pieces of the antigen to be expressed. So the difference between this is in class 1, there was only a little bit of the antigen being presented, but in class 2, since there are 2 peptides, there's a longer piece of antigen being presented in this MHC class 2. This is an exogenously obtained antigen, so this is expressed in low numbers. So we have class 1 versus class 2. So as we said before, MHC class 1s are expressed on nearly all nucleated cells, so most of the cells in the body. This is an endogenous pathway, so these endogenous peptides presents to CD8 cells, so cytotoxic T cells. These MHC1s will have cell-mediated responses. So right here we have the class 1 molecule, and it is made of one peptide, so they're connected all together. But this binding groove is not deep enough for a peptide to come in and bind to it. So we kind of think of the beta-2 microglobin as a peg leg. So before the beta-2 is inserted, the peg leg is removed, so then the 2 sections right here kind of bend over and open up wide enough for a peptide to insert itself. Then the beta-2 microglobin shifts back into place to hold up and close this peptide in. This amino acid sequence has sites at the 2 and 9. So 2 is the amino terminal end, and then 9 is a carboxyl terminus of the peptide. For class 2, this is expressed on antigen-presenting cells, such as macrophages, dendritic cells, and B cells. The difference is this exogenous pathway, which presents peptides to CD4 T helper cells. This pathway in these MHC class 2s will have a humoral response. The T helper cells still need to give permission to the B cells for clonal expansion. The difference between this binding site is these are now two separate peptides. So there isn't much that has to happen for this to open up like class 1 molecules. They have non-covalent interactions. It's open at the end, so it has more of an overhang. So it allows a little bit of extra of those long peptides to hang over the side. So kind of like an extra long hot dog. This can hold up to 13 to 18 amino acids, whereas class 1 is between 8 to 10 amino acids. Unlike the class 1, class 2 molecules are distributed throughout this entire bonding site. So that's the difference between these two things. So like I just restate before, class 1 molecules have a shallow bonding site. So the beta-2 microglobulin has to be moved so that the peptide can bind in the group. Class 2 molecules do not have this shallow bonding site, but they have a long binding groove where extra long peptides and antigens can insert. The beta microglobulin does not have to be removed or put back in since they are two different separate peptides. And that is all, and thank you for watching.